Hi guys, welcome to part 2 of Unit 2. Here we're going to explore how substances move across the cell membrane. So here's where we're going to see all of that time we spent talking about the structure of the cell membrane. Here's where we get a chance to see it in action. So just a quick review, structure and function of the cell membrane. Remember, it's a lipid bilayer that has very fluid qualities. It just puts those phospholipids together, creates that dual layer. As we know, the hydrophilic heads point outwards towards water, the hydrophobic tails point inward. Basically, in water, they're going to orient that way due to the polar nature of the heads and the non-polar nature of the tails. So just as a little sort of cartoon drawing, I have my multiple phospholipids on one side. They all just sort of sit next to one another. Note, not bonded to one another, literally just sit there. And on the other side, I get them pointed in the opposite direction. Again, those hydrophilic heads pointing outwards towards the primary water nature of the outer cellular area, the intercellular layer, mostly water. We also know that the cell membrane is composed not just of phospholipids, but of multiple membrane proteins. These are pretty critical. They have multitudes of functions. We've mentioned some of them already, but we'll go back. So we've got the enzyme piece that you can find within the membrane. We also have transport proteins. Now, transport proteins are going to be involved with actually moving substances across that bilayer. We also have receptor proteins. Receptor proteins are critical when we're talking about hormones. Okay? Hormones have to act at a receptor to signal off the basic chain reaction that occurs to get the outcome that it's there to do. Example, if insulin is going to mobilize glucose or sugar into a cell, that's what it does. It sets off the signal for the cell to suck glucose up out of the bloodstream. For insulin to work to do that, it has to bind to a receptor located on the surface of the cell membrane. If it doesn't bind to that receptor, glucose is not brought into the cell. We also have recognition proteins. These are there, just as they're named, they're there to recognize different cell types. They're also there for self versus non-self recognition, to know if there's something in you that's not supposed to be. Sometimes these have little carbohydrate, almost like antennas, if you will, that sit capped with them. We also have adhesion proteins. Adhesion proteins, just like this, they're going to adhere cells to each other. So they basically help our cells stick to one another. Crossing the membrane. Big new term, we're going to be using it a lot. Cell membranes are selectively permeable. So this new term of permeability, what that means is the ability to cross. Does the membrane allow for things to go across them? Is it Now, by selective, that means that only certain things can cross when others can't cross the lipid bilayer. I like to think of the cell membrane kind of like a bouncer at a nightclub. Okay, hey, you're on the list, you can get in, you're not, you can't. It actually will control what can go in and out. Now, if you're a molecule that's very small, if you're non-polar, and if you're water, you can actually just wiggle your way right through that lipid bilayer with no trouble. You can just kind of bump those phospholipids aside and whoop, right in. Here's some examples right here of some things that are capable of simply just flowing through the lipid bilayer. However, if you're something bigger, if you're polar, if you're big, if you're an ion, so even ions, charged molecules, are charged atoms, these buggers are small, but because they're charged, they cannot cross. Okay, so these guys denied access right here. Okay, cannot cross freely. They would need help, and that's where something like transport proteins come in, and other type of channels that are made out of proteins in the cell membrane would play a role here. Okay, because they can't just, right through that phospholipid bilayer. So how do molecules move across? Molecules move across really in what amounts to I guess fundamentally two ways. Either they diffuse across or they get help, okay, when they move across. So to kind of kickstart that off, we need to get a basic understanding of diffusion and along with concentration gradients. Okay, this is a term, again, we're going to use a lot. The idea of a gradient simply refers to a difference from one area to another. And you could be talking about all kinds of things. So we're going to look at it from the standpoint of concentration differences. Okay, so here's your working definition of it. A concentration gradient, we're talking about the difference in the number of molecules or ions, basically atoms, okay, between two adjoining regions. Okay, so imagine, if you will, if I could set something up on sides, right? If I have a side A and a side B here, okay, if I have molecules all over the place and a couple over here, that's a gradient. There's more on side A, there's less on side B. If they were even, if we were at equilibrium, there would be no gradient. So diffusion, and we've talked a little bit about this, is the movement of these materials from an area of where they're highly concentrated, like in A over here. Here we have our high concentration, to an area of low concentration in the absence of other forces. That means it doesn't require any additional energy. I don't have to worry about inputting anything extra. The simple movement and the natural molecular motion is going to drive this. So just a little visual for you. Okay, here we're going to look at compartment A and compartment B. We're looking at potassium chloride. And you can see that given what we're looking at here is time, your initial condition, okay, diffusion potential exists, so we're starting to have motion occurring. You can see overall movement. Basically, just as molecular motion is happening, these molecules are bouncing, they're bouncing off of each other, they're bouncing off the edge of the beaker that they're in, and they're spreading out. And that will be until you get equilibrium, and all that means is that the concentration over here is equal to the concentration on the other side. This doesn't mean molecular motion stops. Those molecules keep going. It just means that there's no overall change. The concentration on one side is pretty much the same as the other. And what drives this, again, is the inherent kinetic energy of the molecules. Okay, that's still going to drive. One thing I want to know, just because these molecules are moving and my overall movement is from that way to that way, it doesn't mean that that little molecule right there might not go that way. It could. Okay, you're going to get exchange no matter what. It's just, again, what's your overall movement? And when I watch this, the overall molecular motion is going to be from the left side across to the right. Those individual molecular motion, that's completely random. You can't control a single molecule. It's bebopping around, okay? Again, net movement, overall movement is going to be directional. So when I look at this, my overall net movement is this way. However, this little bugger here can still go that way. And diffusion is a critical process. It's absolutely important and the key to the movement of stuff like that. That's super scientific. Okay, you better like have that one down. Of stuff across the membrane and through the cytoplasm. And this goes along with why cells have to be so small. Okay, we have to be able to move things from one end to the other in a very fast speed, and if cells are too big, you have not enough surface area, that surface area to ratio volume is too small, it's not going to get there in the rate that it needs to. There are going to be some things that will affect both the direction of diffusion and the rate of diffusion. When we did the cell size and diffusion lab, what we found out was rate didn't change from cube to cube to cube. The rate of diffusion was the same, meaning the speed at which those molecules were diffusing from outside of the beaker into the cube. What was different was the completeness. But there are things that can affect rate, how we basically determine how fast those molecules are going to move. Okay? Now, the steeper the gradient, the greater the net movement, the faster diffusion is going to happen, which kind of makes sense. You know, when you think about it, if I have, let's say, a very steep hill with a boulder sitting on the top and I let it go, it will hit the bottom and move significantly quicker than if I have a very shallow gradient with my boulder here. Okay, they're both going to travel down, they're both traveling in the same direction, but because one is steeper, it's going to move faster. And this is the same case with concentrations. And those molecules are going to move until equilibrium is reached. And all equilibrium means is that there is no overall change. Okay, so right here I have equilibrium. There is no overall change. However, molecular motion is still occurring. I'm going to have this molecule still could go over here, this one can still go over there. Nothing's going to affect that, but what I won't see is any net change. Another thing, another factor that's going to alter the rate of diffusion is going to be temperature. The higher the temperature, the faster diffusion is going to happen. And that's going to happen because as you increase temperature, you increase molecular motion. And that's going to cause those molecules to move faster, thus disperse a lot quicker. Diffusion is also influenced by how big the particle is. If it's too big to fit through whatever it is that it's passing through, so let's say the membrane or the barrier, if that membrane or barrier is impermeable to the substance because the substance is too big, obviously diffusion can't occur. If they really have to squeeze through, it's going to happen slower. So the size can greatly influence the rate of diffusion, or even just the ability for diffusion to happen, period. Most of the time what we're going to be talking about deals with concentration gradients, actually looking at the movement of substances from an area of high concentration to low. However, bear in mind that gradients aren't just about concentration. There can be pressure gradients. Pressure gradients are vital in our kidney function, in fluid balance, and let's say from not allowing our organs to like swell or tissues to get swollen with fluid. So pressure is a big deal. There's electrical gradients that can occur, so it's not just about concentration. I also want to throw out there that gravity can play a pretty big role in causing a gradient, right? Okay, if I have a waterfall, yeah, heck yeah, water's going to flow downhill. Right? I think that's going to have a pretty significant higher gradient than if I have just like, a flat stream. All right, movement of water. The movement of water across a membrane is so important, and we could still talk about the diffusion of water, and water is going to diffuse back and forth quite often, but it is so important that we actually give it its own term, which I'm going to get to down the road. Part of what moves our water is not just diffusion moving it, but what we call bulk flow. And this is huge movement of one or more substances in response to either pressure, gravity, or other kind of force. So like I mentioned with the waterfall, water falling, okay, down, caused by a gravitational gradient, that's bulk flow, okay. The air, we open up our mouth and really pull in air, okay. If you probably noticed, it's a whole lot easier to bring in air when we have an open airway than we do if you try to breathe through a straw. If you know anybody who has asthma, whenever somebody has an asthma attack and their bronchioles get really thin, okay, and that radius changes, the ability to pull air in makes you have to work a lot harder. It seems like it's a lot harder to breathe. If you want to know what that feels like, literally take a straw and try to breathe through it. So here are some examples of what I mean by bulk flow. Water coming out of a hose, okay, waterfall I've mentioned, inhalation and exhalation of air, that's something that happens a lot. And can you come up with any others? I want you to think about that for a minute. See what you can come up with as other examples of bulk flow. From here, we're going to move into talking about osmosis, but that is a vodcast for another day. Have a great night, everybody. We'll catch you in class later. Take it easy. Bye.